Hi, everybody. My name is Vilma Pérez. I work at the Australian Centre for Ancient DNA at the University of Adelaide. And today I'll talk about environmental DNA, but in the context of ancient DNA. So, first, I just want to acknowledge the Ghana people as the traditional custodians of the Adelaide region where the Australian Centre for Ancient DNA is located. And I also want to acknowledge that in ACAT, we live and work on many different nations' land. So I want to pay my respect to the elders past and present. So the reason why we're talking about environmental DNA today is because in recent years, ancient environmental DNA, or specifically sedimentary ancient DNA, has revolutionized our understanding of past environment, ecosystem, and even human history through different articles like the ones that you can see here. And it has really pushed the boundaries of what we thought it was possible, including this remarkable achievement published in 2022, where researchers recovered DNA that was over 2 million years ago from permafrost samples in Greenland, nearly doubling the age of the oldest DNA ever recovered, which was previously from a mammoth tooth. And this was accomplished because DNA is essentially everywhere we look, not only in organisms, but also in the environment, where it receives the name of environmental DNA. But what is environmental DNA? There are a range of different definitions, and what most people refer to as environmental DNA is the genetic material that can be extracted and studied from environmental samples, like air, water, soil, and sediment, without the need to isolate the target organism first. So as I mentioned here, sediment is included there, and we'll go back to that in a minute. So a key property that defines environmental DNA is that we are typically dealing with very complex samples that contain DNA from many different organisms. And just because of that, it provides a unique challenge in regards to extracting the DNA of that environmental samples because each substrate, so each different type of environmental sample, has specific chemical properties that can make very difficult to extract that DNA. Sorry, I'm just going to go because the animation was wrong. So this scenario is even more complex when we add the element of time on top of that. Even when we're studying a single ancient organism, we know that it's complex. And this is because time is unkind with DNA. So the further we go back in time, the more challenging is to retrieve usable DNA. So due to this challenge of working with degraded DNA, plus the complexity of having a mixture of different organisms in a sample, ancient environmental DNA studies were developed much more later, a bit more than 20 years ago now, as we can see here on the right. With these two articles, the one on the left studying how purple sulfur bacteria were changing in lake sediment over the Holocene, and then the second one on the right, looking into the changes in genetic records of plants and animals over the Holocene and Pleistocene in permafrost samples in Siberia. So that was the beginning of ancient environmental DNA, or in this case, sedimentary ancient DNA, because they were using sediment. So from all the different sources of ancient DNA over the past decade, there has been a growing interest in researching ancient environmental DNA, especially the one from sediment that is present in sediments, sorry. As we can see here on the right, there is an increasing number of publications each year on this topic. And this is because sediment gathers a series of factors that are beneficial for this type of study. So, for example, sediment consists of inorganic and organic matter, including environmental DNA, that accumulates over time, creating natural archives. And the vertical stratigraphy that sediment have allow for the reconstruction of temporal transects of whatever is contained in that sediment. And of course, the sediment helps the DNA preservation when it's because it's oxygen depleted, especially if it comes from cold sediment in aquatic environment. Any caves is shelter from environmental conditions, and it can have stable temperature and humidity. And also, it can collect the genetic signal from different organisms, as we can see here in this diagram. Um, and if we can recover that information from each layer, we can eventually reconstruct the environment for over time. So for example, in cave sediments, we can find information on organisms that have used the cave, including animals and humans, animals that have nested in the cave, like rodents and birds and bats, but also potentially a more distant genetic signal that comes from environmental DNA that has been transported through air or water, pollen, and even dust into the cave. And in aquatic environments, the genetic record in sediment is equally rich and is composed by particles that are brought from the catchment, so the surrounding environment in this aquatic system, like plant material, for example. But it's also composed by autochthonous material, so it's coming from the lake or the system itself through the chemical precipitation in the lake, the biological production from animals living in the water column, for example, like fish, but also organisms that are living in the sediment itself, like microbes. So according to this information that is here, we can find two different types of DNA that have been defined like sedimentary ancient DNA that sometimes we refer to the whole DNA that we can find in the sediment, the sedimentary ancient DNA. So we need to be careful when we're reading articles. So some people call sedimentary DNA to younger DNA that is better preserved in the sediment. And we also have sedimentary ancient DNA that is often older and poorly preserved. So we have ancient molecules, right? And in this category, we can find dead cells or extracellular DNA that's coming from these dead cells that can be free in the sediment or also attached to certain particles in the sediment matrix. And also we have DNA within viable or living cells like cysts, spores, pollen, and even eggs. And these cells might be actively growing within the sediment or in a dormant state, and they're capable of reactivation under the right environmental conditions. But to interpret these genetic records correctly, it's important to first understand the processes that affect that composition of the genetic record in the sediment and also what factors would impact the preservation of that DNA. And all these processes are called taphonomic processes, and they refer to the mechanisms that regulate the production, the transfer, incorporation, and preservation of the sedimentary DNA. And understanding these factors is essential because they play a very important role in how reliable these genetic records are going to be and how we can actually interpret that genetic record. So in this section, I'm going to focus on aquatic environment, as you can see here in this image. But cave sediments are also affected by this type of processes, but this is a bit less understood. So in this figure here, it summarizes the taphonomic processes affecting the sedimentary DNA record, shaped by different environmental and biological factors. And as we can see here, there are several factors that come into play in determining how the DNA is going to be incorporated and buried within the sediment. And it will depend a lot on the conditions of each system, in this case, aquatic system, and different factors like the abundance and the distribution that each organism is going to have in that system, or whether they can form protective covering like cysts and spores because they can be more resistant to certain environmental conditions, and even if their edibility by other organisms, if they're being eaten by other organisms, essentially. So we have primary DNA sources that include direct input from above the ground organisms through decay, shedding, or waste production, for example, urine or feces, as well as from soil and sediment dwelling microbes that are living in the sediment. We also have secondary DNA sources that come from DNA that is introduced through processes like the formation of soil from rock and organic matter through physical, chemical, biological, and even environmental interactions over time. So things like erosion, overland flow, and interflow processes play a key role in transferring the terrestrial DNA into lake systems, which will impact the spatial distribution that DNA from specific organisms have in the sediment, um, but also how diluted that signal is going to be once that DNA is incorporated into the sediment, or even if it will degrade or not. For example, if we have more terrestrial plant DNA, if more terrestrial plant DNA will be expected to be transported to lake, if we have higher soil erosion, and if we have more developed hydrological connections. So we need to take into account those processes when we're interpreting the genetic record. And finally, we have other factors that are playing a role in the DNA preservation, but we'll talk a little bit more about that in a few slides. So what happens to the DNA? So after the DNA molecules are released into the environment, they can go through different processes. For example, we have biotic degradation that happens because extracellular DNA becomes susceptible to nucleases, which, as you know now, they break down the DNA. And this is likely the most immediate cause of DNA degradation in the environment. We also have natural transformation because living cells can take up this extracellular DNA that we have on the environment, even when it's extensively damaged, and integrate them into their own genomes. And we also have abiotic decay, as we already saw in the previous talks. Once the DNA repair mechanism stop after an organism has died, the DNA will start to get fragmented and accumulate chemical damage. And but despite the occurrence of all these processes, extracellular DNA can still persist in the environment. And optimal DNA preservation conditions in aquatic environment would include, as I mentioned before, cooler temperatures, lake with minimal disturbance of sediment layers by living organisms, anoxic conditions, if they're freshwater, and if they have a neutral to slightly alkaline pH in the water lake. But also, DNA can be protected from nucleases by binding to organic components of the sediment, like humic acids, due to the negative charge that they have in the surface, and that can prolong the DNA survival in the environment. But also, DNA can be protected from degradation when it's absorbed onto mineral grains in the sediment matrix, which reduces how accessible the DNA is for nucleases. So this absorption process is crucial for preserving DNA molecules in sediments for thousands of years. So let's just see a little bit more how they work. So different minerals will have different absorption capacities. But different studies have shown that specific clays appear to be the most effective at preserving environmental DNA compared to other components of the soil and sediments. So let's just see an example of one of these. So here we have a smectite clay, which belongs to the 2,1 clay type. And this means that it has two tetrahedral layers surrounding an octahedral sheet in the middle. And in the case of smectite, this middle layer contains different amounts of positively charged ions, and that charge allows organic and inorganic compounds to be inserted into this layer. And this effectively nano-confines and shields the DNA from different factors, including nucleases, that make cause degradation over time. So as you can see here, the DNA that is absorbed onto the clay is still, it can still be degraded by nucleases, but the one that is confined within this middle layer is more protected. So how does the workflow to analyze sediment sample looks like? So we first start by, of course, collecting the sediment sample in the field. And we have a few options here. So if we're working in aquatic system, we need to core this lake or the seafloor. Or we can collect sediment samples from an archaeological excavation, like in figure 1B here. Um, and if we're taking sediment cores, like in figure A, the cores then need to be sampled in a clean lab, following, of course, the strict ancient DNA protocols to avoid contamination. So that means opening the sediment core and then collecting the samples that is usually collected by cleaning the surface of that half core and then collecting the sediment from the middle of the core. And then these samples are sent to ancient DNA facility for the analysis. And once they're there, the sediment samples now need to be subsampled to obtain the material that is going to be used for the DNA extraction and then the sequencing. So the amount of sediment that is going to be collected or subsample will depend on the specific lab protocol that is going to be used, but it typically goes from 250 milligrams to several grams. So it really depends on the protocol that you're going to use and also the target taxa. So if you're working with very low abundant taxa, that you need to use more material to recover more of that sediment, of that DNA. So once the sediment has been subsampled, the first processing step in the lab is the DNA extraction, and then is followed by the preparation of sequencing libraries, because most of the approaches that we're using right now are sequencing dependent. And within these sequencing-based strategies, there are three most used methods. There are metabarcoding, shotgun sequencing, and of course, enrichment with target captures. But we can also apply other methods that are not sequencing based. They are used for quantifying the DNA of a specific taxa or species, like qPCR or droplet digital PCR, that will give us a quantification that we cannot really accomplish by using either metabarcoding or shotgun sequencing. And so as you can see here, this method has great potential, but as in any other ancient DNA research, there are a lot of precautions and considerations that we need to have. So one of the most important is, of course, the contamination control, as in any other ancient DNA research. So we need to prevent the contamination from exogenous DNA material that can go into our samples, and especially when we work with very complex samples like environmental samples, it's very hard to separate what is contamination to what is actually endogenous DNA coming from the sediment. So we need to be very careful of not introducing any potential contaminant. And this contamination control really begins in the field. Um, and we need to follow rigorous protocols that include protective gear such as glove, face mask, and full body suits. Um, if we can do that, sometimes we work in really, really hot environments and it's not really feasible, but we need to protect the samples as much as we can. And one of the ways of tracking contaminants in the field, especially when we're coring aquatic systems, is by the use of either chemicals or DNA-based traces that are essentially applied to the sediment core to assess the extent to which potential external contamination is infiltrating the extracted core. And as I mentioned before, we can apply this to the surface of the core before we sample. And once we open the sediment core and we cut it in half, then we can quantify. If we're using a DNA-based tracer, we can quantify, for example, salmon DNA, if that's the one that we use. We can track it in our samples when we're analyzing them. And if we have a positive detection of this DNA tracer, it means that there is contamination from outside coming into and infiltrating into essentially the middle of the sediment core. So we are aware of potential contamination either coming from the surrounding water or even between layers of that sediment. And another crucial factor that we need to be aware of is when we're creating a list of species from the DNA sequences found in sediment, is having a good reference database. If these databases are not thorough and carefully managed, you might not be able to identify all the species in your samples. Some databases might have contain errors like contaminants or wrongly identified sequences, which could lead to incorrect results. And this, of course, is a problem in any genomic research. But here again, it's really hard to track down when we have those issues because we're working with very complex and highly diverse samples. And a very good example of this, although it's coming from a modern metagenomic study, is this widely reported analysis of the New York subway, which suggested that pathogens such as Yersinia pestis that causes the bubonic plague and Bacillus anthracis, which causes anthrax, were part of the normal subway microbiome. But when these samples, this data, this metagenomic data was reanalyzed with appropriate method. They did not detect the pathogens. So we need to be careful when we're using commonly used software pipelines because they can produce results that lacked a validity of the results. So we need to avoid that by appropriate use of inclusion and exclusion sets, and of course, by validating the presence of, for example, in our case, ancient DNA damage. Another challenge when working with sediment is that the spatial distribution that environmental DNA or ancient environmental DNA has on sediment can be highly heterogeneous, even at a microscale. And this study done by Masilani in 2021 is a very good example by this, where they essentially, as you can see here, they had a resin embedded sediment core, which then they cut to have microstratigraphic resolution of this DNA analysis that they were doing. And then they sample, as we can see here, different points of that sediment layer. And they did a DNA extraction of each one of those points. They were targeting different type of substrates in that matrix. So we had sediment. They also found coprolites. They have some bone fragments. And they realized that they had a different composition of mitochondrial DNA from each one of those samples by depending on the samples that they were analyzing. So, and they also noticed that, for example, the samples that corresponded to coprolite or bone samples were enriched in this mitochondrial DNA. So it really shows the challenge that it is to work with sediment, but also how we can sample that sediment. We need to be careful with that. One key challenge in sedimentary ancient DNA studies is also the potential for vertical DNA movement, so DNA leaching through the sediment, especially in non-frozen environments. So we know that water can move through the sediment profile, and this is less likely, for example, in lakes that are saturated with water, but it can be a possibility in sediments that are coming from examples like caves. So again, this is a great example. This study done by Hale in 2007, where they extracted ancient DNA from sediment dating back to around 3,000 years from two cave sites in New Zealand, in the North Island in New Zealand. And what they wanted to target was the presence or absence of DNA from non-indigenous fauna, like sheep, that was introduced by Europeans. And then also the detecting the presence and absence of DNA from indigenous fauna, like moa, which were flightless birds that were present in New Zealand until, of course, they were extincted. So as we can see here, we have the amount of moa and sheep DNA in each one of those cave sites. And as we can see here for post-European layers, the detection of sheep DNA was correct, but as we can see here, some pre-European layers also contained sheep DNA, which indicated that the DNA was actually indeed moving through the sediment stratigraphy. So that gave us an indication that this indeed can happen. And the best way of addressing this is ideally finding those markers that could indicate this. For example, here in Australia, for a lot of the studies that we are developing, we look for those species that have been introduced after European settlement, um, like sheep or even cow, and we detect them through the sediment core. And if we find them in pre-European samples, we can then assume that the DNA was moving through the sediment core, and we need to be careful how we interpret those genetic records. And then a very important factor that we need to incorporate, or proxy that we need to incorporate into the studies, is classical paleoecological proxies, like the use of pollen or geochemical analysis. And this is because by combining the DNA results with traditional proxies, it can make a more robust paleoenvironmental reconstruction, and it can provide a cross validation of the method. So, for example, here, different studies have shown that pollen compared to fossils of a plant, plants compared to the DNA of plants give different information. Sometimes there is no overlap in that information because DNA is usually give us an indication or information of what is happening in a local scale, so closer to the lake, compared to, for example, pollen that could come from more distant and regional sites, and then is incorporated into the sediment record. And each proxy will be influenced by differences in the biomass production, the distance from the source to deposit, and also the preservation. And for example, in this study done by Parducci in 2019, where they studied past vegetational communities, and they reconstruct these communities from lake sediments in southern Sweden over the last 15,000 years using shotgun method genomics, but also pollen and macrofossils analysis. And they found again that each proxy provided unique insights. So pollen revealed information about both local and regional vegetation. Instead, the plant macrofossils reflected local plant communities, and environmental DNA provided a very strong signal about local plant communities. But by comparing these proxies, the researchers were able to differentiate plants that were growing within or close to the lake from those that were present on a broader scale or region. So, but the DNA offer a more robust indications of the changes in plant communities compared to other traditional proxies. So it added another level of a robust identification. And so I just want to now show you some of the applications that we can use this method. And one of the main ones is paleoenvironmental reconstruction. A good example of a paleoenvironmental reconstruction is this study published by Carr and colleagues in 2022, where the authors recover ancient environmental DNA from permafrost sediment samples in North Greenland. And this landscape, as we can see here in these images, it looks now like a harsh polar desert. But in this formation, researchers had previously found a wide range of plants and insect fossils, but they didn't find signs of mammals, for example. So now they wanted to use environmental ancient DNA to look into the past and reconstruct this biodiversity combined with many other proxies. They didn't just use ancient environmental DNA, but they also used pollen and geochemistry analysis of the sediment and many others. And this was, of course, a great place to look for ancient environmental DNA because it's a cold and dry environment, which is great for DNA preservation as it slowed down its degradation in the environment. So as you can see here, there are different layers within the sediment, this permafrost, and then they took a sample from all these layers and then they reconstructed the environment and how it changed over time using this information. So after they recover the sedimentary DNA from those layers, what we can see here is that they sequence using shotgun metagenomic the DNA that they recover from these layers to obtain as much information as they could to reconstruct what was happening to this part of the world. They first look at animals that they were detecting through time. And as we can see here in axis X, we have the different samples of that sediment record. And then the shades of red indicate how much DNA of different animals they were finding in those samples, from birds to mammals, and even some insects. And they did the same for plants. So we have the different type of plants and their changes through time in axis X. So I'm not going to go into too many details with this study because there is quite a lot of information here. But I just wanted to highlight some unexpected findings in the study. So the DNA analysis revealed more than 102 different genera of plants, so a very high diversity of plants, including 24 that had never been found fossilized in the formation. So they were finding extra information. Then they found 9 animals that included horseshoe crabs, hares, geese, and even mastodon, which is incredible because there was no previous record of mastodons that far north. Some of the plants that they found here and animals are now found in the Arctic environment, but also they're only found, there are others that are only found in more temperate boreal forests. So when they look at all of this information that they collected, they realized that the life in the environment that they were reconstructing 2 million years ago was potentially being developed in a much warmer period in this time of the history of Earth. So the landscape, which is now this polar desert that I showed previously, may have looked like this back then. This is a beautiful illustration that they did for the paper. So I love showing this one. But we can find or gather a lot of information, not just from animals and plants from the sediment, but microbes can give us a lot of information about the environment as well. And this is because they perform essential ecological function. They're really the foundation of long-term ecosystem health and resilience, and then play a major role in determining how an environment is going to recover and what the trajectory is going to be after a disturbance, like, for example, a bushfire. But we don't really know what these microorganisms are doing over time after an environment changes. So we can use ancient environmental DNA to collect information about microbial communities and then trying to reconstruct these communities and how they're changing over time. Another great example of the potential of ancient environmental DNA, especially using microorganisms, is this work published early this year where they study the post-glacial soil formation during the past 23,000 years in Lake Lama sediments in northern central Siberia using ancient metagenomics. And they focus on species, when they were trying to reconstruct this environment over time, what they did is trying to focus on species that show this postmortem damage in their DNA to verify they had an ancient origin. So they also then focused on the most abundant plant, fungal, and bacterial taxa that they were recovering. And they wanted to see the difference between the late glacial period and the Holocene. And as we can see here, here we have the changes of plant, fungal, and bacterial community of the most abundant communities for the late glacial period. And then here, the Holocene. And as you can see, they found significant changes comparing both periods, and they had completely different communities in the Holocene compared to the late glacial. So what they did next is reconstructing this ecosystem, and they have this again beautiful illustration where we can see how the ecosystem and the soil dynamic changes over time according to the data that they found in the study. At the top part here, it shows the changes to plant communities over time. And at the bottom here, it shows the changes of the most abundant soil microbial communities, which were dominated mostly by bacteria and some fungal taxa. Their functions are highlighted, as you can see here, in circles with different colors. And the size of the circle represents the importance of each element cycling process for at each in each period. And on the left here, we have changes occurring over time during the late glacial period, so between 23 and 12,000 years ago. And on the right here, we have the changes during the Holocene in the last 12,000 years ago. So very briefly, during the late glacial, there was a strong weathering of the bedrock, and there was an early colonization of plants in this area, and this vegetation then developed towards a shrubby tundra, so it changed over time. And in the soil, carbon, sulfur, and arsenic cycling was higher during this early colonization of these plants. And then on the right, during the Holocene, Pinacea, so a type of plant, invaded this area, and an early Laryx forest or different type of forest was then established, and it was then replaced by spruce, which led to an increase in iron cycling here in the soil, and the start of the formation of potsoil soil development. So, but now these changes in the soil properties enable that Laryx came back and established again as a forest. And during this period, the nutrient cycling in the soil was dominated by nitrogen cycles. So it's a lot of information that they could recover about the plants, but also about the interaction that these microbial communities, for example, rhizobium communities could have with these plant communities, and how these two types of communities impacted one each other when they were establishing and changing over time. So it really shows the potential that this method has, but also the complexity that that data can has as well. So here we have another study that was looking at phytoplankton changes in response to climate in polar ecosystem. And what they wanted to see was they wanted to use sedimentary ancient DNA records that were covering roughly the last 1 million years from the Scotia Sea in Antarctica. to understand the changes in phytoplankton communities over time. So for that, they reconstructed the eukaryote composition using enrichment, so hybridation captures, using specific markers for the communities for phytoplankton in this case. And they found records of diatom and chlorophyte sedimentary ancient DNA dating back 500,000 years ago. And they could actually reconstruct the oldest marine eukaryote sedimentary ancient DNA record today. They also, apologies for the resolution of the image, but they could actually also collect information about the ancient DNA damage that those communities had because they were using hybridation capture instead of metabarcoding, for example. And they saw that there was overall an increase of that DNA damage the older the sample was. And overall, they concluded that warm phases were associated with high diatom abundance and a significant shift from sea ice to open ocean species around 14.5 thousand years ago, following meltwater pools 1A. So, and finally, it's not all about microbes, but also a little bit about hominins and ancient humans. So we have this study that was done in the Denisova cave in Siberia, where they use again hybridation capture to enrich in mitochondrial DNA from fauna and hominin in this site to reconstruct the changes of these communities over time. And one of the very impressive things here in the study is that they analyzed DNA from around 700 sediment samples that were collected in a grid manner from layers that were dating around the Pleistocene. So we can see here the number of samples and the effort, the sampling effort that they had. And this is very important, especially going back to how heterogeneous the sediment record can be. So unfortunately, we could collect samples from one side and not detect the species that we're looking for. So sometimes, if budget allows, we need to expand that sampling effort to be able to pick up that in a more robust way. And here we can see a simplified version of what they found here for the fauna and the hominin that they detected. And so they retrieved ancient fauna and hominin mitochondrial DNA from 684 and 175 samples, respectively. So for hominin, as you can see, a very smaller proportion of samples compared to animal DNA was found. And they found the earliest evidence of hominin mitochondrial DNA is of Denisovans, as we can see here in red. And it's associated with early and middle Paleolithic stone tools that were deposited approximately 250,000 to 170,000 years ago. Then Neanderthal mitochondrial DNA first appeared towards the end of this period here in blue. And then there is a turnover of the mitochondrial DNA of Denisovans that coincides with changes in the composition of faunal mitochondrial DNA. And there was also evidence that Denisovans and Neanderthals occupied this specific site repeatedly, possible until or after the onset of the initial Upper Paleolithic, when modern human mitochondrial DNA is here in yellow, is first recorded in sediment. So again, it really shows you not only the potential of the method, but the resolution that it can have in this type of data. So these groundbreaking studies have largely been conducted in the Northern Hemisphere regions, but the Southern Hemisphere, especially places, for example, like Australia, is vastly underrepresented, with the exception of New Zealand, which most of that has been led by Jamie Root. He has done an excellent effort in ensure that New Zealand is represented in this global sedimentary ancient DNA record, as we can see here. And as many other places in the Southern Hemisphere, at the Australian Centre of Ancient DNA, we're actively working to change that, to have a better representation of this part of the world. And we have different type of projects being developed at the moment using a variety of samples from sediments or even middens. And we have, for example, some projects that are collecting hundreds of sediment samples from caves nationwide to help build a predictive model for DNA preservation in caves that could allow us to find hotspots of ancient DNA more effectively. But we also have other projects where we're working with indigenous communities in archaeological sites to look at different things like the presence of their old ancestors to environmental changes. And finally here, well, of course, you know of SPAAM, but there are other international communities that can really help you when you're working with ancient environmental DNA, and especially sedimentary DNA, like the Sedimentary Ancient DNA Scientific Society, which is going to start their seminar series soon in August, actually next week. So I invite you to sign up to these communities and read about this. Thank you for your attention, and I just want to acknowledge people at ACAD, University of Adelaide, and also Centre of Excellence where I'm working, CAI.